All right. So I have been tasked with explaining all of inflammation in 15 minutes or less. So we will see how this goes. So I am going to talk a lot about kind of general concepts of our current understanding of how methamphetamine may cause inflammation. And then at the end, I'll talk a little bit more specifically about some of our work in the gut. So this will kind of be more of a general overview, hopefully to get everybody on the same page. And hopefully you'll hear me echoing a lot of the themes that have been brought up earlier in some of the other talks. So let's see if we can, I guess I have to do it this way. All right. So, the answer of where does methamphetamine cause inflammation is likely everywhere. But there's a few different compartments that have been the most highly studied. And so that's what we're going to focus on today. So I'll talk a little bit about the brain with the caveat that I am not a neuroscientist. And so do keep that in mind with my explanation. I'll talk about methamphetamine effects on blood immune cells. I'm not going to talk about endothelium or cardiovascular because that's already been beautifully described earlier. And then at the end, I'll talk about the role of the gut in this whole inflammation landscape. And so one of the reasons that neuroinflammation has been probably the most studied inflammatory site with methamphetamine is that it's thought to contribute to the neurotoxicity. And so studies, both in vitro studies as well as many animal studies, have had some consistent themes that have come out of them. So one is that methamphetamine increases production of IL-6, TNF-alpha, and IL-1 beta. And when you look across all of the studies, those three cytokines are constantly the ones that are coming up. And so those really seem to be three key cytokines in this whole inflammation and methamphetamine relationship. This inflammation will lead to glial cell activation, and this is thought to be in part due to glutamate pathways as well as reactive oxygen species, so oxidative stress. And eventually this persistent neuroinflammation can lead to neuronal injury, and that I'm not going to talk about because that is far outside my area of expertise. And so how does this neuroinflammation occur? So one contributor are cytokines, which I mentioned. And so how this occurs is that methamphetamine will actually induce production of the cytokines from the glial cells, again, IL-6, TNF-alpha, and IL-1 beta. These cytokines will then activate microglial cells and can, in some of these animal models, lead to this reactive gliosis, which is a response of these glial cells to injury that occurs like during stroke and other injuries and so forth. And so these cells are constantly activated. They then produce more cytokines and chemokines. And this kind of just perpetuates this inflammation and eventually can lead to neurodegeneration. And using animal studies with IL-6 knockout phenotypes in mice, that phenotype has actually been protective against damage, neuronal damage, and apoptosis from methamphetamine, further showing the importance of cytokines in this cascade. So I also mentioned glutamate and oxidative stress. So methamphetamine will increase extracellular glutamate concentrations. This glutamate can increase the reactive oxidative species. And these will then change the cell redox status. And that can trigger transcription factor activation that then will lead to production of inflammatory cytokines. Separately, there's actually direct signaling from the glutamate receptor. I think it's receptor 5 or something along those lines, that will increase production of IL-6, TNF-alpha, IL-1 beta, and IL-8. And this occurs through a PI3 kinase, AKT pathway, and NF-kappa B activation, leading to this cytokine production. So all of these different kind of things converge together to just promote this ongoing inflammation. And so what was brought up earlier was the importance of the blood-brain barrier. And I think this is kind of an even step back further from the mechanisms I just described as one of the first things that occurs. So the blood-brain barrier is supposed to protect the brain, right? It keeps all the toxins and other bad things from getting into the brain to this privileged site. And so there's an endothelial layer that has these proteins in between that keep the cells together to create this barrier, these tight junction proteins, claudin, occludin, things like that. So methamphetamine shows up and these proteins start to either degrade or they're not produced as well, and this barrier starts to break down. And so what this allows is this transmigration of inflammatory cells, most commonly inflammatory monocytes, which will then differentiate into microglial cells. And these are actually really important in the pathogenesis of neuroAIDS. And so this further contributes to this instigation of neuroinflammation. So it leads to this breakdown of the blood-brain barrier. Again, we'll bring up this oxidative stress. So these are data from a study using human brain microvascular endothelial cells exposed to methamphetamine. So in the top panel, you can see that they're measuring with a fluorescent marker the reactive oxidative species. And the second line from the top, the kind of black one, is the methamphetamine. The top line is their positive control. And then when they add the methamphetamine in the presence of an antioxidant, they do not see that increase in ROS. The bottom panel shows the barrier function, right? So they're measuring the transendothelial resistance. And so you can see again, sorry, there's a little from my screenshot there. You can see again that with the methamphetamine, this function of the barrier is significantly decreased when the methamphetamine is added in the presence of an antioxidant. They do not see that decrease in barrier function. So oxidative stress seems to be a critical mechanism for blood-brain barrier dysfunction. All right, so I'm going to move on to the immune system. So the effects of methamphetamine on the immune system are really interesting because it seems to simultaneously cause inflammation that I've just been describing, but it also causes a relative immune suppression. And we know that's because people who use methamphetamines are at higher risk for many infections, not just HIV, but they have worse outcomes with things like histoplasmosis and other respiratory infections and all these other infections. And a lot of that is thought due to immune dysfunction and immunosuppression. And so the arm of the immune system that's most well studied with methamphetamine is the innate immune system. And so a lot of people have focused a lot on macrophages. And so macrophages are the cells that will phagocytose bacteria and other pathogens. It presents it to B cells and T cells for the adaptive immune system. It also has antimicrobial properties and things like that. And so methamphetamine will actually inhibit the phagocytosis capacity of macrophages as well as dendritic cells, also can inhibit antigen presentation, and can induce pro-inflammatory cytokine production. So it's making them cause inflammation, but also stopping them from doing their protective function. It also has been shown to decrease natural killer cell function, which is another main immune cell, increases cytokine production, as I mentioned. And all of this ends up resulting in reduced immune function. The adaptive immune arm, which is your T cells and your B cells, are much less well studied. So the few studies that are out there are focused largely on T cells, and they found kind of this nebulous functional impairment, details of which have not really been well described. And then a lot of alterations in T cell populations, specifically CD8 and CD4 ratios. Again, this T cell dysfunction in particular is thought to be one of the biggest contributors to this increased susceptibility of infections. All right, so we'll move on a bit to the gut because it's my favorite topic. And the gut is really, really important when we think about this because a lot of people don't think of the gut this way, but it's actually your body's largest lymphoid organ. So it is the place where about 80% of your total body lymphocytes reside. So it is a critical immune organ. We just think about it for, where our food goes, but it's actually a really important immune organ. And so this is a study done with Steve many years ago, where we looked at rectal inflammation specifically in people who had recently used methamphetamine, so who were UTOX positive. And so what we found actually is that even after adjusting for HIV serostatus, for age, sex, and receptive anal intercourse, there was increased inflammatory cytokine production in the rectal mucosa in people who had recently used methamphetamine. We simultaneously looked in the blood, and we did not see those changes. So there seems to be some sort of compartmental inflammation induced by methamphetamine. And so in other studies, it has also been shown, kind of similar to what I described about the blood-brain barrier, the methamphetamine affects the gut barrier. So, right, so the gut is, a tube that's full of bacteria. Essentially, that's what the gut is. And so the barrier there keeps the bacteria in the tube from getting into your bloodstream where they're not supposed to be. This barrier can break down. And so in the top graph there, is from a rat model, the HIV transgenic rat model. And they had rats self-administer methamphetamine, and then they looked at gut barrier function by measuring a sugar that goes through. And you can see that the methamphetamine in the black bars increases the amount of translocation, so the amount of stuff that goes through that barrier, basically. And that this is worse with the HIV transgenics. So these are data actually from my lab using M-study samples, showing that there's actually increased markers, this is in humans, of intestinal barrier dysfunction. It's FABP2, or intestinal fatty acid binding protein. And so what's kind of interesting here, so these are HIV negatives and these are HIV positives. Oh, you can't see my pointer. So the purple and the green are the HIV negatives, and the red and the yellow are the HIV positives. And so you can see among HIV negative, there seems to be increased of this intestinal inflammation or intestinal barrier dysfunction in the methamphetamine. It's not statistically significant, but you can see it there in the graph. What you can see is in the people who do not use meth, between those who are HIV negative and positive, there is a significant increase in this gut barrier dysfunction. And this is known, this has been shown in other studies. In the people who use meth, when they go from HIV positive to negative, you don't see that difference. Probably because they already had dysfunction from the meth, so the addition of HIV is not really causing any additional thing that we can measure, at least in these plasma markers. All right, so what does meth do to the gut? So these is studies, again, from my lab, where we actually isolate gut immune cells from human colon. So we get either surgical explants or surgical remnant tissue, or we do endoscopic biopsies of the mucosa. We isolate the immune cells, or we culture the entire tissue in the presence or absence of methamphetamine, and then we use either flow cytometry or mass cytometry to look at the cell phenotypes. So what you can see here are untreated, so just looking at the cells themselves. PMA ionomycin is a positive control for immune activation. And then methamphetamine is there on the right. So these are spade trees. So the way to kind of look at these is that they all look different. So something is happening. Each node is like a cellular population. The size of the node has to do with the number of cells, and the colors have to do with the relative expression of a given marker. In this instance, it's CCR6. The important thing to keep in mind, or to notice here, is that the methamphetamine looks different than the untreated. So the meth is doing something, and it's different than this kind of classical immune activation. So the meth is doing something unique. And when we look at the cell, oh shoot, it didn't come up. So when we look at the cell populations that are different, so this kind of there on the bottom looks red, and then the cluster at the top. When you actually look at what those cells look like, they're T cells, and more specifically, they're Th17 cells. And so it seems as though meth is doing something to Th17 cells in the gut. And what that is, we're trying to figure out. And so you can see again here, in kind of a more standard flow cytometry plot, that with increasing concentrations of meth, we get more T cell activation, shown there by higher expression of HLA-DR. All right, so knowing that there's some inflammation, we are also interested in what meth may be doing to the gut microbiome. And so this is, again, a study using M-study samples, looking at the gut microbiome in about 400 men who have sex with men, who are meth users and not meth users, and who have HIV and who do not have HIV. And so when we look, just looking at meth use versus non-meth use, there are microbiome differences that you can see. And so if you look at the taxa bar plots over on the right, like look at the two big rectangles, you can see that the one on the left and the one on the right do not look the same. Therefore, there are microbiome differences. So when you look at kind of, we do some modeling and so forth, we can see specific taxa that are different between people who use meth and people who do not. And what they tend to be are a lot of these, what are in other studies considered inflammatory or bad bacteria. So we see increases in Porphyromonas, which has been widely associated with cardiovascular risk. We see decreases in certain Faecalibacterium, which are considered good bacteria, healthy bacteria. And so it seems as though the microbiome changes that we're seeing are reflective of inflammation. So how might this be occurring? So we've already kind of described how meth may be causing gut inflammation. But one thing that a lot of people have not looked at when you look at what meth does to the body is, remember, there's all those bacteria that live in your gut. They are a part of your body. So what is meth doing to those? And so this is a study I like to affectionately call bugs on drugs. And it's like my favorite project we're working on right now. So we're actually growing these candidate bacteria that we identified in our study in the presence and absence of meth, which is a bit of an ordeal to grow some of these because they're all anaerobes. And so when we look specifically at Porphyra monas, grown in the presence or absence of increasing concentrations of meth, you can see that the kinetics of the growth look the same, but the plateau is different with meth. And so that's considered the carrying capacity of the bacteria. And so what that may mean, and this is, again, we're kind of early on, so this is right now occurring in like a plate, a tube. But what it may mean is that in a limiting resource setting, once that bacteria has kind of reached its maximal nutrient absorption, the addition of methamphetamine allows it to continue growing when the other bacteria stop without meth. And so you can imagine that in the gut, where there's a lot of inflammation and resources may be limited, that advantage may allow those bacteria to grow better. And when we look at some of the other bacteria that did not change or did not change as much, we don't see any difference with the addition of meth. So it does seem to be something that may be specific to bacteria. And so right now we're actually growing these bacteria and isolating the supernatant in the cells, and we're doing metabolomics, because the other important thing about the gut is all these bacteria also make a bunch of metabolites, and those will actually control a lot of the immune system and so forth. So seeing how the methamphetamine may affect the metabolite production of these bacteria is also critical to kind of putting this all together. All right, so how do we put this all together? So this figure is from a paper, and it's kind of summarizes kind of our current conceptualization of inflammation in HIV. So the inflammation is driven a lot by just HIV production, viral replication, loss of regulatory T cells, and this concept of microbial translocation or barrier dysfunction in the gut. So I already mentioned that meth contributes to T cell changes in T cell populations. Meth contributes to microbial translocation, barrier dysfunction. And what I didn't talk about is that meth actually contributes to viremia. And that's been shown in studies from us and studies from a lot of other people, that even when people take their antiretroviral therapy, they still have difficulty with viral suppression. All right, but what remains is the big black box, is how does this all happen? And so a lot of the mechanisms to tie this together are completely not understood. And I think that's one of the areas that, I think we can kind of think about ways that we can answer some of that and move the research further. I can tell you my kind of conceptualization of how the gut may play a role in this based on work we've done is that I showed you that meth may directly affect the bacterial metabolism and growth, and that this may confer a survival advantage in an environment like the inflamed gut. Separately, methamphetamine can cause T cell activation, specifically of Th17 cells. That can contribute to barrier dysfunction, and all of this comes together to cause an inflamed gut. That then contributes to dysbiosis, contributes to microbial translocation, and that then leads to inflammation. The other kind of interesting thing with our finding about the Th17 cells is that's the cell that is preferentially infected with HIV, particularly at mucosal inflammation, or mucosal transmission. And so if there is an increase in these cells in the gut as a result of methamphetamine, that may be a biological explanation for the increased incidence of HIV transmission. All right, so to kind of summarize all of inflammation, what I talked about is that meth causes neuroinflammation in part via glial cell activation, cytokine production, the oxidative stress and glutamate signaling. Increased glutamate contributes to that as well. And that damage to the blood-brain barrier is really probably a driver of all of that. It affects macrophage function and T cell function in the blood. And that it does seem to induce some gut inflammation. It has been associated with gut dysbiosis, and the mechanisms we're still trying to work out. So I'll leave you with some lingering questions that are at least still on my mind and things to think about. So one thing that really bothers me, and this is partly because we have data that shows this as well, and I cannot explain it, and it is not what my hypothesis was, so it's bothering me. So we see this localized inflammation in the brain and the gut and the endothelium. But when you look at systemic markers, the data are not consistent. And we've seen that now in 2 different studies where there's maybe some increase in the systemic markers in people who use meth, but it's never usually statistically significant and it's not consistent. And when you look at other studies that are in the literature, when they're looking at inflammation, it's usually in a specific area and it's not always, like plasma markers. And so why is it that we see all this inflammation in these different places, but we don't see kind of these systemic markers? And are we just looking at the wrong markers? Because we know the inflammation is there, but I don't think we're capturing it well. What is the role of the sympathetic and parasympathetic nervous system in regulating this inflammation? How can that maybe connect our brain and the gut? Are interventions that target this oxidative stress, which seems to be critical, sufficient to reduce some of this inflammation and improve clinical outcomes? And then I'm personally just very interested in the gut-brain microbiome axis. And so are those communications contributing to regulating inflammation? And so I think I went over my 15 minutes, but I've left enough time. So these are my founding sources and the wonderful people that either do this work or help me do this work. And hopefully I got through all of the information and you now completely understand all of it.
That was an excellent presentation. One thing in the model that I will certainly add, because I think it's sort of like a common thread, is the endothelium. Every single organ you put there, including the lung, is covered with endothelium, and the endothelium sort of serves as a master regulator for many things. I don't know about the microbiome, but I imagine there has to be an interaction between, the microbial flora and endothelium.
Yeah, there absolutely is.
Right? So I also agree with your proposal that compartmentalization of these biomarkers, these cytokines, is going to be critical. I mean, we're sampling from blood, but we don't know whether those cytokines reflect activity in the brain.
Yeah, I think.
And so I think that is going to be a critical play, because that by itself can provide us also with unique opportunities to sort of dissect what's happening on each kind of organ and how to connect the dots. Last but not least, if we were to add the lung, and I'm hoping we can add the lung to your brain lung axis, the pulmonary circulation is different from the systemic circulation, which is what serves the brain and the gut. So, for example, in the setting of hypoxemia, you have vasodilation in the systemic circulation. In the lung, you have vasoconstriction. So right there, that tells you that the endothelial cells in that compartment behave much differently. So there's also some underlying genetic differences that I think as we apply deep phenotyping, we can see that not all endothelial cells are equal. And sort of like, to understand this, I think we're going to have to think creatively about how to look at each compartment.
Yeah.
And ultimately also at a higher scale, how to integrate those things. So I think thinking about like, more of a precision medicine where we can sort of integrate metabolomics with single cell and microbiome, I think this is the way where we can really understand what's happening here.
Yeah, I mean, I agree 100%. And I think that the last point that you brought up, the metabolomics, may be one of those keys, because, when we look at biomarkers, most of what we look like are cytokines and other proteins. And it's really the metabolites that are actually doing a lot of the communication between these compartments. And so I think we may actually see a better signal looking at metabolomics in some of these settings than just looking at standard biomarkers. And I also think that, that to be able to do this, look at kind of this from the different compartments and bring them together, that's the beauty of this meeting with everybody with different expertise, because the endothelium is absolutely super important. I just don't know much about it.
One thing,
Same thing with the brain and
correct
so
me if I'm
forth.
wrong, but one of the biggest products of microbiome is butyrate, right?
Yeah. Well, it's one of them. Yeah.
It's
It's
one
one
of
of the ones that's very well studied. And so people
Right.
focus on it a lot when they're doing like targeted metabolomics. But if you
Right,
look at
exactly. So
untargeted,
this is
yeah.
why, I mean, not knowing much about it, that's the one thing that pops out. But
Yeah.
the reason it pops out is that, it also, as you know, it influences histone acetylation, which is a huge mechanism in regulating transcription. And there's data coming out, at least in the field of pulmonary hypertension, that the butyrate production can be a critical determinant on pulmonary endothelial transcriptional activity,
Oh, wow.
because the butyrate can influence histone deacetylation, and that by itself can influence the entire landscape of genetics. So I think that's another very important. So if that's the case in the lung, which is, it's
Yeah.
closer to the gut, we can imagine what's happening at the level of the brain.
Yeah.
Circulation.
Yeah, at least one or two of the bacterial genera that we saw decreased with meth are butyrate producers. So.
Fantastic talk. A quick question on the sort of trajectory of this. Sounds like you've studied a lot with heavy meth users. Do you have any sense of earlier meth use and see if that helps you segregate those who are going to get into
So
trouble?
we did in the paper trying to look at kind of like a dose response, if you will, of certain bacteria compared to frequency, self-reported frequency of meth use. And for some of them, we did see almost a dose response in the increase. That's the only point we've looked at that. A lot of the other studies that we're looking at right now, we are kind of focusing more on the heavy meth users just to see what the phenotype is, and then later go back and look at, okay, what degree of meth contributes to this phenotype.
Can I follow up on that? You may have mentioned this already, does the route of abuse influence that response?
We have not looked at. The only thing we've done in these studies is we've excluded rectal use for our obvious reasons, since we're looking at the gut, and that would be a little unfair. But beyond that, smoking versus injection, we specifically haven't examined, but I believe, Steve, you can correct me if I'm wrong, that there are very few injectors in M study. So the majority of these are either smoking or inhaling.
So your preclinical model requires meth to be present along with the bacteria. So are you thinking that the meth in the gut is what's driving it, or are there systemic changes happening that then influence the gut microbiome?
So now you're going to get into like my little pet hypotheses. So what I didn't mention is the mouth, right? And so we know that meth causes problems in the mouth, meth mouth. So and I have another project that's actually specifically looking at the oral microbiome in the exact people that were in that gut study, using paired samples from that exact visit, again, from the Glorious M study. So there are separately studies where people have looked at samples from the mouth and the gut over time, done shotgun sequencing, and actually traced bacteria using like SNPs, basically, so using single variant sequencing, and have shown that there's translocation from the mouth to the gut. A lot of the bacteria that we are seeing in the gut that we think of inflammatory are often, in our ID world, thought of as oral bugs. And so what I think may be happening specifically with meth is that these bugs may be exposed more to the methamphetamine in the mouth. When the people are using the drugs, they translocate to the gut. That's not a normal environment for them. They change the whole environment when they show up. Like they show up in the neighborhood doesn't know what to do. And that just triggers this kind of inflammation cascade and kind of starts that dysbiosis. That is my personal hypothesis. I have no data to back that up, but I'm trying to get there.
Yeah, and just one comment on the model, like Th17 cells are really good at disrupting blood-brain barrier, like they have been shown. So that could also be an extra
that's interesting.
factor. Yeah, so
that's interesting.
course, the methamphetamine needs to get absorbed to travel, so it really travels across the body. And I imagine that in some places, endothelial cells may be more apt to suck it up. I mean, there's
Yeah.
receptors in the membrane known to actually allow methamphetamine to enter the cell and interact with receptors in organelles like the endoplasmic reticulum, for example. So I think again, also the endothelial phenotype,
Yeah.
different organs is actually a critical.
Yeah. Yeah. No, that's a really good point. I like that. Everybody hungry?